Hi everyone, welcome to the Chapter 16 video. Today we're going to continue with our discussion about genes and genetics. While last chapter focused on the replication of DNA, now we're actually going to hone in or focus, I couldn't decide what to say, on exactly how DNA works as genes, okay? So you guys have known for a long time that DNA is the genetic material and that your genes are encoded in DNA. But exactly how does that work? So this is going to be sort of an overview chapter that sort of bridges us to our next chapter, chapter 17, which will discuss how genes work in more molecular detail. The things I want you to get from this chapter are, first of all, I want us to review the central dogma, okay? And if you don't remember what that is, that's okay. We're going to go back over it. The second thing that I want us to go over is exactly what the genetic code is. Exactly how does DNA represent a code for genes? And then the third thing I want to talk about is the role that mutations have. Because mutations, while often thought of as being problematic, they're also a source of genetic variation and ultimately how populations evolve. So they're very, very important in this conversation. So to get us started, let's first take a look at the central dogma. Now, you have seen this diagram before. This was shown to you in chapter 1, but let's go back over it. The central dogma essentially is a thought process that describes how genes code for proteins. So back in 1953, remember that Watson and Crick discovered, or at least put together, synthesized a bunch of ideas to formulate the structure of the DNA molecule. And at that time, remember, we knew that DNA was the hereditary material. So Francis Crick, when looking at the molecule of DNA, he proposed that the bases acted as a code, sort of like words in a sentence. But he also knew that there must be an intermediate step. He knew that DNA could not all by itself be a code. And the reason that he knew that was he knew that there was all this stuff that was going on out here in the cell. But DNA never, ever leaves the nucleus. It stays in the nucleus. So it only stood to reason that in order for DNA to be a code that could exert action out here in the cytosol, but DNA didn't leave the nucleus, there must be some sort of messenger traveling between the information in the DNA and the cytosol of a cell that tells the cell what to do, that basically relays this genetic information to the rest of the cell. Excuse me. And RNA was definitely proposed as an intermediate, because what folks did know about RNA at the time is that you could find RNA inside the nucleus, and you could also find RNA out here in the cytosol, okay? So in looking at RNA a little more closely, what we found was that there was an enzyme called RNA polymerase that could copy DNA and turn it into RNA. And it functions very similarly to DNA polymerase in that if we open up a molecule of DNA, so here I'm going to draw a molecule of DNA. Actually, let me do it a little closer. So we're going to draw us a little molecule of DNA just like we have been. And remember that according to the semi-conservative understanding of DNA replication, we know that these two strands that are in opposite directions from one another, they pull apart, right? So we're going to have these strands pulled apart. And what RNA polymerase is going to do, so here's the other strand, is it uses only one of these strands, okay? And RNA polymerase is going to come along and it is going to make a strand of RNA. This time my orange means RNA. The same way that DNA polymerase made DNA. Now, how is it different? We're going to go into the specifics of this process, but essentially what's going to happen is this single strand of RNA is going to go off by itself. And this single strand of RNA can be messenger RNA as well as other types of RNA. Once this strand leaves, then the DNA re-anneals, which is a fancy word for sticks back together. Okay? So what researchers at the time knew was that they could find RNA in the nucleus. They knew that they could also find RNA out in the cytosol. And they knew that there was an enzyme that was capable of copying DNA into RNA through a process known as transcription. So these folks hypothesized that this RNA was functioning as a messenger molecule that could move between the nucleus and the cytosol, carrying the genetic message between the two locations, okay? And they decided that if this were true, mRNA should be complementary to the DNA strand. So what they decided was that if RNA polymerase was copying DNA to use it as the genetic message, then what we should see is that if you have a strand of DNA, then mRNA should complement that strand of DNA. And so what researchers did early on to confirm this hypothesis was they isolated RNA polymerase and they made some fake strands of DNA that were composed of nothing but T's. So they were able to synthesize a strand of DNA that was just T's. And they said, look, if this RNA is functioning like we think it is, and if RNA polymerase is functioning like we think it is, then what will happen is, this is the color I was using, then the RNA molecule that results should be nothing but A's, and it should be this single-stranded molecule that can float around. And that's exactly what they found. So essentially, at this point in the late 1950s, we're beginning to understand that RNA is this messenger molecule, okay? I do have a note down here that RNA polymerase is similar to DNA polymerase, but does not require a primer to begin synthesis. We'll talk more about this in chapter 17, okay? So the results of this experiment where we confirmed that RNA is a messenger molecule allowed Francis Crick to really develop his central dogma idea, okay? So the central dogma is based around two concepts, transcription and translation. Let me go back up to our picture up here. So what the central dogma essentially says is that, let me go back to my black. The central dogma says that the DNA hereditary message in the nucleus is copied into RNA, and this is a process known as transcription. Okay, sorry, I'm just trying to spell that out. So when you think about the word to transcribe something or the word transcription, that usually means that you're making an exact copy in the same language of something. So here we're talking in the language of nucleic acids. Yes, we're going from DNA to RNA, but we're still in a nucleic acid language. So we're transcribing this message exactly into another nucleic acid type. The next thing that Crick proposed, is that this mRNA, this messenger molecule, leaves the nucleus and travels to the cytoplasm, where it is then translated, let me erase this, it is then translated into protein. So this process of going from mRNA into protein is called translation. And when I think about remembering this term, when you translate something, you're moving from one language to another. So you're moving from the language of nucleic acids to the language of proteins, which remember, those are encoded by amino acids, right? So we've changed languages here. We've translated, okay? So we've got transcription, which copies the DNA into mRNA. The mRNA can then leave the nucleus and travel to the cytoplasm where we have ribosomes. Ribosomes take this message, this mRNA message, and they turn it into proteins via this process of translation. Now, the interesting thing is that proteins are ultimately what does the cellular work. And Francis Crick was able to understand that and say, you know, it's the cellular work, things like enzymes and structural proteins. It's the cellular work that results in the traits that we're able to observe. So let's go back to this slide here. So what Francis Crick was saying was that the idea that DNA codes for RNA, which then gets translated into protein, he understood that these proteins explain the relationship between genotypes and phenotypes. Your genotype is in your DNA. It results in a protein, and that protein ultimately results in what you see as a phenotype or a trait. I'm going to put trait/phenotype. Let me give you a specific example to show you what I mean. Okay, so here we have two different mice. These are actually the same species of mouse. One lives in an area that is wooded. So you can see that this is a really dark mouse. It lives in the woods. Okay, this mouse lives at the beach. And you can see that its coat color is quite different than the color of this wooded mouse here. Now, how might that happen? Well, it turns out that there is a gene called the melanocortin receptor gene, okay? It's often abbreviated the MCR gene, okay? So the melanocortin receptor gene is the receptor that initiates pigment production, okay? So basically the melanocortin receptor allows this guy to make plenty of melanin, which is the pigment that results in this mouse's dark color. So here we have the DNA sequence for this gene, okay? This DNA sequence gets transcribed into RNA here. So you can see that this is a complementary copy of this DNA. And we do have one difference here. We'll talk about that in a second. Instead of having T's, we have U's in RNA, but otherwise, this is a complement that you would expect. Now, what we're going to talk about in this chapter is that this mRNA directly corresponds to a code for amino acids that go into the protein for this melanocortin receptor, okay? So this code can be translated into these amino acids, and then that ultimately results in the functional melanocortin receptor protein. So basically, this genotype here results in this phenotype here. Now let's look at our little light-colored mouse. Here we have a different genotype. Notice that here, this letter A is different than this letter G. So this adenine is a mutation away from this guanine here. So when this gene is transcribed into RNA, we get a different mRNA molecule. This mRNA molecule is then translated into a different series of amino acids. Notice this amino acid here is a cysteine, and that's different than this amino acid here, which is an arginine. What that does is it results in a non-functioning melanocortin receptor. And ultimately, that means that pigment does not get made in a proper amount. Now, it turns out that's okay. If you're a beach mouse, this is a nice camouflage. So this phenotype is actually adaptive and was propagated throughout this population because these guys were eaten less, right? But what I want you to understand is how a change in the genotype can result in a change in phenotype, okay? So let's see here. Now, this idea of DNA going to RNA, and I can't write this enough, so I'm just going to write it again. This idea of DNA to RNA to proteins, and that the proteins ultimately resulted in phenotype. It led to an understanding of biology that was sometimes summarized as the one gene, one enzyme theory. And you could substitute that to mean one protein, but this was back in the early days of understanding this. So basically, one gene would code for one particular protein. Back up here, we have one gene here, and it's coding for the melanocortin receptor. Over time, we've grown to understand, though, that there are a few exceptions to this central dogma theory. It is generally true that this is how information flows through your cells, okay? But there are a few exceptions. So first thing I want you to know is that some genes are transcribed into RNA, but they're never turned into proteins. So the process stops right here. There are some RNA molecules that function, that have cellular functions all by themselves. So like we're gonna learn about tRNA, which is involved in translation. We're gonna learn later on about rRNA, which is involved in the structure of ribosomes. There are other RNAs that we'll talk about later in the course called microRNAs. And we may even talk about something called long non-coding RNAs. So these are various RNA molecules that are transcribed from DNA, but we don't turn them into proteins because these all by themselves have functions within the cells. So that's one modification to the central dogma theory. Another modification to the central dogma theory comes from our understanding of viruses, okay? There are some viruses that don't use DNA as their genetic material. Instead, they use RNA as their genetic material. And so what happens in these viruses is, here's an example of one. These viruses are called retroviruses because they do things backwards. And so what these viruses do is they infect cells. So here's a virus infecting this cell. It's coming on in. Its genetic material is being released. This is the genetic material right here, and it is RNA, okay? Then, along with this RNA, this virus comes prepackaged with a special enzyme, that's it right there, called reverse transcriptase. And I'm just going to abbreviate that RT for short. And what reverse transcriptase does is instead of going from DNA to RNA, no. What reverse transcriptase does is it does it backwards. It takes RNA and it transcribes it into DNA. And this is an unusual thing that only happens with some viruses, okay? But up until we discovered these viruses, we thought that every living organism transcribed things in the DNA to RNA direction. And the reason this is important is there's some human viruses that are retroviruses that are really important for us to understand. Some that you may have heard of would be HIV, okay? Also, the virus that causes chickenpox or herpes, which is, no, not chicken, is it chickenpox? The herpes virus I know is a retrovirus. I want to say that the chickenpox virus is, but I'll have to look that up. But for sure, these two are retroviruses that start out as RNA and work backwards to DNA. So there are a few modifications to this central dogma theory, but in general, know that the central dogma of DNA codes for RNA, which codes for proteins, which ultimately results in phenotype, is something that I will want you to know. I will want this hammered into your brain. And I tell you that because when I was working at University of Southern Mississippi and I was teaching graduate students, I was always shocked at the number of graduate students that could not recite the central dogma to me. and I find that unacceptable. So my students will for sure understand the central dogma. All right, moving on. So I told you that DNA codes for RNA and RNA codes for proteins. So how do we get from DNA to RNA to proteins? How do we get there? We know that somehow these are acting as a code, but what exactly does that code look like, okay? So that was the next big question in our discovery and understanding of DNA. Scientists were really interested in understanding how RNA coded for proteins. And most scientists figured that somehow the nitrogenous bases, the A's and C's and T's and G's that are in DNA, which are then turned into, let me do this way. In RNA, we don't have A's and C's and T's and G's, do we? In RNA, what we have are A's, C's, U's, and G's. So remember that in RNA, uracil is substituted for thymine. But we figured, we being scientists at the time, figured that these bases, the order of these bases, must function like words in a sentence, and that these bases must function as the code that determines the protein, okay? So in thinking about that, our good friend Francis Crick, remember the same guy who was involved in understanding the structure of DNA, he thought about like, well, what would this code look like? What if every individual base, an A or a T or a C or a G, what if each of these coded for an amino acid? Well, the problem with that hypothesis was that we only have 4 different bases available to us in our DNA genetic code or our RNA genetic code. Also, I'll put TU. So we only have 4 bases, which means that we would only be able to get 4 amino acids out of a code where each individual base represented an amino acid. So Francis Crick knew that that couldn't be the case. So what if it was every 2 bases? What if it was a sequence of every 2 bases, like AC, AG, AT/U, and then you could have CG, CT, et cetera, et cetera. If you work this out, it comes out to be 4 to the second, right? Is that right? Yes, that's right. It comes out to be 4 to the second mathematically. 4 to the second, or 4 squared, as you guys know, that would ultimately end up in 16 amino acids. In other words, if we used every 2 bases, if we said this coded for an amino acid, this coded for an amino acid, there are only 16 possible combinations that you can make of 4 different bases. 16 combinations would code for 16 amino acids. Now let's remember how many amino acids we use to form proteins. We use 20 amino acids to make proteins. So ultimately, a code that included only 2 of these bases to refer to an amino acid would also be insufficient. So Francis Crick did the math and he said, well, what if we used 3 of our bases? What if we used every 3, let me erase here. What if every 3 of these 3, what if we went 3 by 3? How many possible combinations could we come up with? Well, the way to figure that out mathematically is to take 4 to the 3rd, okay? If you do that math, that comes out to be 64. And you can satisfy yourself that that is true. 64 is more than enough variety of combinations to cover a code for each of our 20 amino acids, right? So Francis Crick proposed that the genetic code must consist of every 3 bases corresponding to an individual amino acid. And I'm not going to make you memorize experiments in this chapter, but do know that this was verified experimentally, right? And we've actually broken these codes. So we now know that the RNA bases, or the DNA bases, because DNA codes for RNA, these bases form words that code for amino acids. How long is each word? It is 3 bases long, and we call these 3 bases that form a word corresponding to an amino acid, we call those codons. So this is a codon here. This codon corresponds to this amino acid, which happens to be an amino acid called methionine. These 3 here, they code for this amino acid right here, which is isoleucine. These 3 here, these code for an amino acid called serine, okay? So this is the genetic code. Oops, excuse me. This is the genetic code. This is how we know, or how we go from being a DNA molecule to an RNA molecule to a protein molecule. Over time, what we've experimentally been able to do is to figure out exactly what the code is for each and every amino acid. So if you count here, there are 20 different amino acids. Let's just verify. 1, 2, 3, 4, 5. Don't count that. 6, 7, 8, 9, 10, 11. Don't count that. 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23. We have some that are repeated here. Let's see here. Arginine is repeated. Cysteine must be repeated somewhere. Let's see here. I'm losing track. We're going to talk about how these are repeated. But you can see that we have every single amino acid here is in fact covered. All right. We also have some additional codes here, the start codon, and we have 3 stop codons. So let's basically break down this genetic code here. The way that you read this chart is you start here. First base U is in red. Second base U is in blue. Third base U is in green. The code UUU corresponds to a code for an amino acid called phenylalanine, excuse me. But notice that phenylalanine can be UUU or it can be UUC, all right? And so that's how you would read each and every one of these. So let's look at proline here. Proline is a C, and a U, okay? So here we go, that's proline. But this is also proline, this is also proline, and this is also proline. So you can see that there are 4 different codons that code for proline. Here there were 2 that coded for phenylalanine. So let's look at some of the features of this chart, okay? So first of all, I want you to notice that we have something called a start codon. That start codon is right here, okay? And it also corresponds to, oops, excuse me, to the amino acid methionine. So the start codon is AUG. Well, what do I mean by start codon? This is the signal, the genetic signal to start translating into amino acids. So each and every protein starts with this AUG codon, okay? This is the genetic signal that says, hey, we're going to start transcribing a protein right here. This is the beginning of the sentence, okay? So every protein originally begins with the amino acid methionine because that is the same thing as the start codon, all right? Sometimes that methionine is chopped off later, but every protein begins life with the methionine at the beginning, okay? The next thing I want you to notice is that on our genetic code chart here, we have 3 stop codons, 1, 2, 3. So there are 3 stop codons. These stop codons do not correspond to an amino acid, but they are the signal that says to the ribosome, hey, we're going to stop translation, okay? We're going to stop translation. Okay, I have a whole list here of things I want to tell you, so let me make sure. Okay, the third thing that I want to point out that I've already kind of pointed out is that our code is redundant. What does that mean? Well, notice that I told you that we had a couple of different codons that equaled phenylalanine. We have 4 different codons here for proline, 4 for serine, 4 for alanine. Remember that if you use 4 to the third, if you use a triplet code, you have 64 possible combinations that you can make of the 4 bases that we have, right? So that means that some of these amino acids have more than 1 codon associated with them. And we're going to see that that's actually a good thing later on when we talk about mutations, okay? All right, let's see here. What else do I want to tell you? I think I must have given myself a second slide because I knew I would run out of room to write this for you. Okay. So the code also is unambiguous. Now, what do I mean by that? Ambiguous. So that means that each codon is specific. Let me describe that a little more closely. Let's take a look at this codon here, okay? Let's look at GCU. GCU specifically codes for alanine. GCU will never ever mean another amino acid. It will never mean serine. It will never mean glycine. GCU always means alanine. It is unambiguous. Yes, there are 4 different codons here that also mean alanine, but GCG here, this one, this bottom one, always means alanine. It will never mean valine, okay? It is unambiguous in that way. The next thing I want to tell you is it is not overlapping. So let me show you what I mean by that. So I'm just going to take and make a little short strand of RNA, okay? So I'm going to make up a short strand of mRNA code, okay? And so I'm going to start out with the start codon, and then I'm going to, let's see here, let's do GCU, GAU, GGU, U, oops, GA. So if we look up these codons, AUG codes for methionine, which is our start codon. So I'm going to put MET. GCU is right here. It codes for alanine, which I think is abbreviated ALA. GAU codes for aspartic acid, which is ASP. You don't have to memorize these. If I ever ask you to do this, I will give you a chart. GGU codes for something called glycine. And UGA is a stop. Now, what I mean by non-overlapping is we go AUG, then we move on to the next 3 in the sequence, GCU. So what we don't do is we don't go AUG and then go UGG and then go GG. You see what I'm saying? The code is non-overlapping. We take a chunk of 3 and then we take the next chunk of 3 and we don't do any sort of like sliding down here. We take the next chunk of 3. There is no overlap here, okay? So essentially, we say that we have frames that are called reading frames, and these frames are locked in. Okay, and what I wrote down here, maybe this is a little bit easier for you to understand. Once the frame is locked in, each set of 3 bases counts as a codon. Okay, let's see if there's anything else I wanted to tell you guys on here. Okay. The last 2 things, and I'm going to erase just a little bit here because I've made a bit of a mess. The last 2 things that I want you to know about this genetic code, and this is really interesting, and it really is another piece for life, another piece of evidence for life evolving once on the planet. The code is universal. And what that means is that this set of codes, UUU, UUC, et cetera, this code is true in every organism. I'm going to put all organisms on the planet, okay? This code goes for all organisms on the planet. So if you are a bacteria and you have a genetic code that for GAU in bacteria right here, that codes for aspartic acid in bacteria, just like it codes for aspartic acid in people, okay? And then finally, let me erase all of this. The last thing that I want you to know is that the code is conservative. Okay, so notice that if an amino acid has more than 1 codon, they start with the same 2 first letters. So for example, we'll go back here, we'll look at threonine. Threonine has 4 different amino acids: ACU, ACC, ACA, ACG. I'm sorry, it's got 4 codons. Notice that they all begin with A and C. And the reason that that's important is if you have an ACU, okay, and you have a mutation in this last one, it doesn't matter because you're still going to end up with threonine as your amino acid. So this minimizes the effects of mutation, okay? The wobble in this last letter minimizes the effects of mutation. So while the first 2 are the same, the last one can wobble around, and it gives us some protection against bad mutations. Remember when I said redundancy was a good thing? This is what I meant. And we'll talk about this effect a little bit later in the lecture, okay? So let's practice doing some translation. So here we have some DNA, okay? And we have what's called the non-template strand and the template strand, okay? So the template strand of the DNA sequence is the one that's transcribed. Interestingly, the template strand, we only use 1 strand of the DNA to be transcribed into RNA. So if we're going to use this template strand here, then we are going to transcribe this using our complementary base pair rules. So we would, let me go back here. So T pairs with A. So now we've got an A on our RNA molecule. A pairs with T in DNA, but it pairs with U in RNA. C pairs with G. And you could go on and on and on to get what your mRNA molecule looks like. Now, I want you to notice that your mRNA molecule should be almost exactly the same sequence as your non-template strand, okay? And so with the exception of U's and T's, okay? That's always going to be a difference. So sometimes you will see the non-template strand also called the sense strand, and you'll see the template strand called the anti-sense strand. And people often struggle with this concept, but the deal is that this strand, the non-template strand, makes sense because it's an exact copy of what the mRNA looks like. The antisense strand or template strand is the opposite, okay? It's the opposite set of complementary base pairs. Don't let that confuse you. I don't know if your book does that, but I know that other textbooks will use these terms sense and antisense, okay? Now, if you had an amino acid chart in front of you, you would be able to take this mRNA message and look up each of these codons, okay? And you would be able to translate these codons into amino acid sequences. And it is something that I almost always ask students to do on exams. I will always give you a codon chart. You do not have to remember these codons. It's too many for you to memorize. So if I ask you to do this exercise, I will always give you a codon chart, okay, so that you can go from mRNA to amino acids with no trouble. But what I'd like for you to practice is I'd like for you to practice transcribing this template strand here into mRNA, and then use the codon chart that's in your book or in the previous slides from this lecture to translate this into a protein, to an amino acid sequence, okay? And practice doing that for yourself. If you are correct, if you get this right, and if you want to know the correct answer, you can email me and I'll be glad to send you that correct answer. If you get this correct, then you're in good shape because like I said, I always ask students to do this on an exam. All right, so be sure that you can practice transcribing and translating sequences. So now that we understand how the genetic code works, it's a little bit easier for us to understand the effects of mutation. And so what I mean by that is if we have a base pair substitution, what we're doing is we're changing codons, aren't we? So if we accidentally insert an incorrect base pair, we have changed codons. And this is called a point mutation, where we've inserted an incorrect base pair at a single point on the DNA, okay? Now these mutations, we're gonna come back to this idea, mutations can have three different effects. They can be beneficial, they can be neutral, or they can be harmful, but the word we like to use in biology is they can be deleterious. In other words, they delete things. They cause problems. Okay, so not all mutations are bad. Let's go back up here. I want to go to my mouse picture. I'm going to erase. So let's think about what happened with this mouse situation. What we had going on here, let me make this just a wee bit bigger. All right, what we had going on here was a point mutation where this guanine, this is a letter G here, this guanine, a mutation occurred and an incorrect base pair was inserted during DNA replication. So this guanine has been converted to an adenine, okay? We've gone from a G to an A. So this is a great example of a point mutation. Now, what was the effect of this point mutation? Well, if we scroll down, we can see that this originally translated from GCG into RNA, GCG, which tells me that this is the non-template strand. Okay, so here we've got, transcription into, sorry, I'm saying this all wrong, guys. Let me back up. Okay, don't let me confuse you. Let me start again. This started as G. Then in this situation, it's turned into an A because we've had a mutation. Here, we have, this is a template strand, not a non-template strand. I was misreading it. This is a G, this is a C, this is a G. If we transcribe this according to complementary base pair rules, we have C, G, C, okay? If we look this up on a codon chart, this codon corresponds to the amino acid arginine, okay? Now let's look over here. We have the code A, C, G, because we have a mutation here. If we transcribe this into RNA, that transcribes into U, G, C, because of complementary base pair rules, okay? This, if you look this up on a codon chart, corresponds to the amino acid cysteine. Notice this is a different amino acid here than this amino acid here. The substitution of cysteine actually makes this protein, the melanocortin receptor, or MCR, a non-functional protein, meaning that this guy cannot make pigment. No pigment, but no pig, okay? Now, you might think that that would be a bad thing, and if this mouse lived in the woods, it maybe would be a bad thing, because if this mouse lived in this habitat, then this mouse here, would be really visible to predators in a dark wooded environment. But because this mouse lives at the beach, where it's up against light colored environments, the sand here is light colored, you can see that this mutation was actually beneficial, okay? It's beneficial if the mouse lives at the beach, but it's harmful or deleterious if this mouse lives in the woods, isn't it? Because it would be more visible to predators. So mutations can have different effects depending on the environment in which they occur. All right, so let's go back and look at these point mutations a little more closely. We're going to look at some examples of where base pairs have been inserted inappropriately at a specific point into the DNA, resulting in a mutation. What might the outcome of these different base pair problems be? So there's different types of point mutations that can occur, and I'd like to walk you through each of these types. So this is the original sequence that we're going to use in our example, okay? So you can see that this is the original sequence of the non-template DNA, okay? The non-template DNA. Notice, I know it's non-template because it looks just the exact same as this mRNA molecule, of course, with the exception of U's being present instead of T's. So TAT is UAU. But otherwise, these are the same, right? If you were to look up these codons on a codon chart, you would find that these codons corresponded to these amino acids. So you would translate this into this here. So this is the correct situation. Now let's look at what happens when some point mutations occur. One type of point mutation is called a silent point mutation. Remember how I said that the codon chart is redundant? And I said it was conservative. And what I mean by that is the first two letters of any codon typically code for the same amino acid. There's wobble in the third letter. okay? Meaning here we have had a mutation that went from a T to a C, right, in this example. But it turns out that a mutation here doesn't change the outcome because both UAU and UAC, both of those are codons that code for tryptophan, for TYR, or no, that's tyrosine, TYR. So we've had no change, even though there's been a mutation, a point mutation in the DNA that was transcribed into RNA, it didn't change the translation. This is what is called a silent mutation, because even though a mutation has occurred, it has not resulted in a change in our amino acid sequence. No amino acid change. And that means that there's really not an effect on the organism, okay? Now, there's another type of point mutation that can occur. This one's called a missense mutation. Okay, so here, what we've done, let's look at the second codon here. The original DNA sequence was TGG. That got transcribed into UGG, which gets translated into TRP, okay? But if we mutate here, this TGG to TGT, this is an example where we don't have a lot of wobble in this particular sequence. So a mutation here does make a difference. If you look this up on our chart, TGT gets transcribed into UGU, which goes from being TRP, which I think is tryptophan, to a CYS, which is cysteine. This is a different amino acid. A missense mutation codes for a different amino acid. Now, what the effects of a missense mutation are is unclear. It depends on the protein, because if this amino acid is chemically similar to this one, and it has like similar positives and negatives and all that, the change in the overall protein function might not be that great. Or the change could be really large. If this is a positive amino acid and this is a negative amino acid, it could really end up making a difference in the function of this particular protein. So if we get a change in our amino acids, then that change might be beneficial, it might be neutral, or it might be deleterious, might be harmful. We don't know. It depends on what the change is. Okay? Let's look at another possible point mutation type, and that's called a nonsense mutation. In a nonsense mutation, what happens is a mutation occurs. So here we've gone from TGG to TGA. That codes for a stop codon. So you can see that basically translation has stopped. We stop making amino acids because that's what this codon tells us to do. This ends up causing a shortened protein or a shortened polypeptide. We'll put short protein. And this is almost always harmful or almost always deleterious, okay? You can imagine that if you just stop it before you're supposed to, that's usually going to be a problem. The last one that I want to mention is also almost always going to be harmful, and that is a frame shift mutation. Give you guys time to write it. Frame shift mutations happen when you're not substituting one base for another, you accidentally add one in. So if I typed in the fat cat ate the rat, and I added in a letter, let's just say I add in an extra T, then I would have Do you see what I've done there? If I'm breaking this up into groups of 3, E-R-A. Do you see how I've shifted the frame by adding in an extra letter? If I accidentally deleted a letter, that would be a problem too. What if I got rid of this letter H? Then suddenly my frame would be this. All of this makes no sense whatsoever, does it? Etc. You guys can play with this all you want to. So a frame shift is when you have the addition or deletion of a nucleotide, and it shifts all of the reading frames to different triplets, just like I showed you here. The result is basically a bunch of gobbledygook that doesn't make any sense. You can see that once you get here, the only one that's the same as the original is this one. Once you get here, everything from here on out is different than the original. And this is going to be very harmful in terms of making a very messed up protein, okay? Okay, so I accidentally went over this earlier, but we talked about how mutations can be beneficial, they can be neutral, or they can be deleterious. And whether they're beneficial or deleterious sometimes depends on the context. So I showed you in this case where we had a non-functional melanocortin receptor because we had a missense mutation. This is a missense mutation resulting in a non-functional melanocortin receptor. And it turns out this is beneficial in this environment because this guy lives at the beach and he blends in. But it would be deleterious if this white mouse or tan mouse lived in the woods, wouldn't it? Let me show you another example in humans. So I wanted to show you an example from a human disease called sickle cell anemia. Sickle cell anemia disproportionately affects people of African descent. So we're talking about our African American population, our Black population if you're not from America, if you're from the continent of Africa. But if you are of African descent, then you are far more likely to have this mutation than other populations. So what is sickle cell anemia? Sickle cell anemia is a condition in which red blood cells are malformed. These are normal red blood cells. These are red blood cells that have the deformed shape resulting from this genetic defect. And you can see that they're kind of sickle shaped. They're kind of like this, oops, like that. And the problem is when they're misshapen like this, they don't pick up oxygen properly and they don't flow through blood cells through blood vessels properly, okay? So you can imagine that these would get all clogged up in your little capillary vessels and you would be unable to oxygenate your tissues properly. So people that suffer from sickle cell disease often become short of breath during exertion, or sometimes they'll even have organ failure problems because they're unable to properly oxygenate all of their tissues. Now, what causes sickle cell anemia is a single point mutation, and this is a missense mutation situation where we've accidentally substituted one base for another base. So this is the original sequence that would result in a normally shaped blood cell. This is actually part of the gene for hemoglobin, which I'm sure you've heard of. Hemoglobin is the protein that binds oxygen inside of your blood cells. So this is part of a normal hemoglobin gene, okay? But what happens in sickle cell anemia is that that normal hemoglobin gene has suffered a mutation. So here, the DNA sequence is CTC or GAG, but here it's CAC or GTG. Do you see that we've had a change? We've substituted this T for this A. And when we do that, if we go through the process of transcription and then translation, we go from a glutamic acid, amino acid, to a valine amino acid. And that causes misfolding of the hemoglobin protein. Okay, and so the hemoglobin becomes non-functional, doesn't work right. Non-functional. So you would think that this is a very harmful or very deleterious situation, very harmful, because folks like that have this, they have problems, okay? But here's the thing. It turns out that people that have sickle cell anemia are slightly more resistant to malaria, which on the African continent is a very harmful disease. Basically, it is a unicellular parasite that exists inside of your blood, and it gets inside your blood cells. But malaria can't really infect sickle blood cells the way it can normal blood cells. So individuals that have sickle cell anemia actually tend to survive malaria. They don't really, or they don't contract it the way other people do. So depending on where you're living, sickle cell may actually be a little bit beneficial. It's not totally beneficial, right? But it turns out that the harm caused by being unable to oxygenate your blood very well is actually not as harmful as being infected with malaria. Now, I'm very much oversimplifying this situation. Some of you may have had family members that you've been told have the sickle cell trait. And if you have questions about that, I'm happy to answer those questions during our class time. Okay, so the last thing that I want to mention is, up until now, I have been talking about point mutations. And remember that point mutations, those occur at a single point on the DNA molecule. Single point on the DNA molecule. But we do have such a thing as chromosomal mutations, okay, where entire chromosomes become damaged or altered. So I'm going to put entire chromes become damaged or altered. And remember that a single chromosome contains lots and lots of genes. So when we talk about point mutations are only going to affect at most one gene, right? Because they occur at one spot on the DNA. But a chromosomal mutation can affect many genes because chromosomes have tons of genes on them, don't they? So let's take a look at some different types of chromosome level mutations. These are pretty easy. So basically, you can have a situation where a chromosome becomes damaged and a whole portion is deleted. So here, can you see that we've deleted this region of the chromosome here, and it's been repaired, but this little guy here gets lost. So these genes become lost. That's usually going to be very harmful to an individual. If it was like a baby that was being born with this, or at least that cell, that would be very harmful for. Here we have something called a chromosomal inversion, where we have genes A, B, but perhaps at some point this chromosome has become damaged and it was repaired, but this section was flipped upside down. Sometimes that makes a difference and sometimes it doesn't, okay? Here we have a section of a chromosome that's been duplicated, where we have 2 copies of the A and 2 copies of the B. Again, whether or not this makes a difference, it depends on the context. Here we have 2 non-homologous chromosomes. These are non-homologous. And what we have here is part of this chromosome being traded with part of this chromosome. And again, whether or not this makes a difference depends on the context, okay? So just like with point mutations, these can have beneficial, neutral, or deleterious effects, these large-scale chromosomal mutations. Sometimes, like here, it might not make a difference at all if you have a change, if you invert these genes, or sometimes it might. It just depends on the context. For example, in plants, having multiple copies of genes can be good. I have here. In plants, it can be good, but in animals, it can be bad. So it just depends on what you're talking about. The last thing that I wanted to mention is that unlike point mutations, where we have to actually sequence your DNA to know whether you have a mutation, we can actually, oops, oh, come on. We can actually, there we go. We can actually see chromosome level mutations using microscopy, because we can visualize chromosomes, right? So this is an example of a picture of chromosomes called a karyotype. So karyotypes are just images of chromosomes where we've taken a picture using a microscope, and then what we do is we line up homologous chromosomes with each other to form these karyotype diagrams. This is a karyotype from a breast cancer cell of a human. And you can see that it is filled with chromosome level mutations. So for example, in this breast cancer cell, we have on chromosome 3, it looks like, let me give a light color here. On chromosome 3, it looks like we have a deletion here and a deletion here. We also have 3 copies of chromosome 3, okay? I'm sorry, this is chromosome 2. On chromosome 3, it looks like we've actually gotten some extra genetic material from somewhere, and again, we have 3 copies. Chromosome 4, we've either lost information here or we've gained information from somewhere. So you can see that this breast cancer cell is riddled with problems, whether we have extra copies, whether we've lost information, or what have you. When you have an abnormal karyotype like this with abnormal numbers of chromosomes or abnormal types of chromosomes, there's a name for that. It's called aneuploidy. And I just wanted to introduce that term to you. It is something that we explore more in a genetics course, okay? So the big things that you need to take from this lecture are, first of all, you need to make sure that you understand the central dogma. Make sure that you understand the genetic code and how it takes 3 bases to equal 1 amino acid. Oops, the genetic code. And then 3, I want you to know the different types of mutations. Okay, there we go. All right, and so this is the end of chapter 16, only 1 video for this chapter. We'll pick up next week with chapter 17. In previewing chapter 17, if you're watching this in the fall of 2020, the next week, which begins, I think, October 26th, we're gonna only focus on chapter 17. It has a lot of information. So next week when you have your quiz, it will only cover chapter 17. Again, that's if you're watching this video in the fall of 2020. All right, if you have any questions, please let me know and I'll talk to you soon. Bye-bye.